Hi everyone, thanks for coming. My name is Bianca Brown and I'm a grad student at Brown University. And today I'm going to talk to you about evolutionary patterns in a community of small mammals in Kenya. We live in a microbial world, and even though we're constantly bombarded by microbes, there tends to be a mechanism which host filters their microbes. So microbiomes of hosts tend to be distinct from their environment, particularly in mammals. And I'm interested in how this host filtering sort of results in a pattern of microbiomes across host populations and species and genus and genera, all the way up to phyla. And I sort of got interested in this when I first saw this Lay et al. 2018 paper. And essentially, the group showed that microbiomes across multiple individuals on multiple species of mammals sort of cluster by diet. You see, this is a PCOA plot, and it's showing multiple individuals from different orders, and each color represents a different diet type. And you can see that the microbiome of this mammal community clusters by diet. I'm showing the same PCA plot, and essentially, under that diet differences, Lay et al. Showed that there are differences in microbiome composition across order. This structure of the microbiome across orders sort of led to this evolution of people studying microbiomes of multiple species, of multiple individuals across species, and starting to ask questions about how microbiomes clusters across multiple species. And from this study, we found out that microbiomes tend to be species specific. In 2016, this idea of a microbiome being species specific sort of evolved. And what that means, if microbiomes are species specific, closely related species should have more similar microbiomes than distantly related species. And that should sort of result in this mirroring of the microbiome host phylogeny. What I'm showing here is a schematic of the phylosymbiosis theory that attempts to explain the eco-evolutionary relationship between hosts and their microbiome. And the idea is, up on the upper left side, if you look at multiple individuals from different species, you can have clustering of their microbiome in multiple different ways. So you can have the two extremes, where on the left, the right, you can have the stochastic clustering, where there's no species significance of the microbiome. On the left, you can have the species clustering of the microbiome, where individuals of the same species sort of cluster together. And the idea is, if the individuals of the same species are clustering together, you should see sort of this congruence between the host phylogeny and the community distance, and the microbiome community distances. And we started seeing a lot of studies exploring this idea of microbiome mirroring host phylogeny. This is a wonderful study by Amada et al. That was done earlier this year. And we see she explored multiple species of primates, a microbiome of multiple species of primates. And we see these distinct clusters based on species and based on the type of primate they were. So the old world cluster, the lemurs, and so on. You can see this distinct cluster based on phylogeny group. But a big problem, or sort of issue, this is a beautiful study, but these studies that tend to sort of look for what are called these phylosymbiotic patterns tend to be compounded on multiple levels. And what I mean by that, for example, in the Amada study that I just showed you, New World and Old World monkeys are never going to be found in the same space. So their microbiomes are shaped by different environments. So yes, the evolutionary history should be driving it, but we can't really tell if geography has some influence on the microbiome. And a lot of these big studies, like the Lay et al. Study that I showed at the beginning, they sort of incorporate zoo animals and captive animals and laboratory systems. And we know that these animals tend to have eroded microbiomes. So I wanted to understand this phylosymbiosis concept within a community of animals that are interacting and are sort of being exposed to similar environments. And to do this, I sort of co-opted a system across an exclusion system across a rainfall gradient in Kenya. And what I'm showing you is a map of a research site, and each dot within the research site consists of exclusion plots. Within these exclusion plots, the control plots to the right, which is your left. Yeah, the control plots to the left, and the exclusion plots are to the right. And these 1 acre plots, essentially what happens, we build fences, you exclude large mammals, and when you exclude large mammals, you sort of create a havens for smaller mammals. So you see an increase in the population abundance, and you also see this species guild of mammals, small mammals, sort of explode due to the exclusion of larger mammals. And because we have this explosion of this species guild of mammals, we can start asking microbiome questions about how interspecific microbiomes are structured in this community of multiple species under similar environmental conditions. And these species of animals are also from multiple different orders. So you get this very rich phylogenetic evolutionary history from this community. So I wanted to test the phylosymbiosis theory concept within this community of small mammals. And my research question started with, are intraspecific microbiomes more similar than interspecific microbiomes? I also wanted to look at if there is congruence between microbiome community distance and host phylogeny. And I want to ask other questions, such as, are there phylogenetic signals associated with other measurements of the microbiome that are not part of the phylosymbiosis concept? And are host traits, such as weight, influencing or associated with microbiome diversity at any point? For all the data that you're going to see, we did 16S RNA metabarcoding of fecal samples. The numbers at the bottom are the sample sizes. And in total, for this talk, we used 109 samples. So for the first part, my first question, are intraspecific microbiomes more similar than interspecific microbiomes? We started testing the first part of the phylosymbiosis concept. And I'm hoping to see this clustering of the microbiome, based on species. And we do see that. So we see the clustering of the microbiome. So what I'm showing you is an NMDS plot of Bray-Curtis distances, and each color represents a distinct order. The red are the elephant shrews, which is from Macrocyllidia, the hares, the hyraxes, and the rodents. So you see this strong clustering. When you look into within Rodentia, you see this strong clustering by species. So across the entire community, you see microbiome clustering based on species, confirming that the interspecific microbiome, so a species microbiome, individuals from the same species microbiome were similar than individuals from different species. So we could take off the first half of this phylosymbiosis concept. So the next question I wanted to ask is if there was congruence between microbiome community distance and host phylogeny. So this is the second part. And we sort of expected, if we saw this strong species cluster of the microbiome, it should sort of translate into this congruence between host phylogeny and microbiome community distance. On a whole across the entire community, what I'm showing here on the right is the host phylogeny, and I'm using a cytochrome B tree, and on the left is the Bray-Curtis community distance, the mean Bray-Curtis community distance. And using a Mantel test, which tests the correlation between both matrices, we see that across the community, the entire community, we have some congruence between the host phylogeny and the microbiome community distance. However, within Rodentia, we have a lot of crisscrossing, and that signal is that congruent signal is lost. So to summarize the first half, is there congruence between microbiome community distance? Yeah, we see significant congruence between microbiome community distance across borders. And we saw degrees of phylosymbiosis, where I wouldn't say the entire community is showing a phylosymbiotic pattern, but we did see degrees of it. And we need to think about this idea that microbiomes can be species-specific without showing a phylosymbiotic pattern. So not because a microbiome is species-specific, you're going to see it predicting the host phylogeny. So the second half of the talk, I wanted to focus on, is there a phylogenetic signal associated with any other aspect of the microbiome diversity? And is there a correlation between a trait and microbiome diversity, particularly in Rodentia? What is driving all those crisscrosses that we saw in the plot that I showed? Here, I'm showing one measurement of microbiome diversity, the Shannon Diversity Index. On the left, this is the phylogeny that I showed in the beginning, and the mean Shannon diversity plotted as a trait on the phylogeny. And I attempted to check if there was a phylogenetic signal associated with Shannon diversity, and we saw that there was none. The bar chart just shows the diversity index for each individual. And we saw that there was no phylogenetic signal associated with Shannon diversity within this community of small mammals. And I wanted to really dig into rodentia more because, as you remember from the previous class, that there was a lot of crisscrossing in that group. One thing we do realize in this community of rodents is that they vary drastically by size. The smallest individual we have, the smallest species we study is around 4 grams, and the largest ranges from like 90 to 120 grams. So as you can see, there's a lot of variation in size. And we know that size have been shown to be correlated to various microbiome diversity indexes. So what I'm showing you on the left is Shannon diversity indexes regressed against weight, which is our measurement for size. And we see there's a strong correlation between the Shannon diversity index and all the other diversity index, alpha diversity index that we measured in this community and size. On the left, I'm showing microbiome composition. On the right, I'm showing microbiome composition correlated to size at the bottom. So what you're seeing is that the y-axis here is a Bray-Curtis distance from a PCOA pot, and what you're seeing is that species that cluster together tend to be more similar weight than species that are not clustered together. And this is showing that size is driving a lot of the variation that we're seeing in the microbiome. Even though we weren't able to show a phylogenetic signal, we did see that size is strongly correlated across with microbiome diversity measurements within this community. And the fact that size is a strong, I guess, predictor of microbiome within this community, microbiome diversity within this community, it doesn't mean that it's a random trait. The microbiome isn't random. Size is correlated with a lot of physiological processes, especially in rodents. So it's probably like a precursor to understand how physiology is influencing microbiome composition. So to summarize, within this community of small mammals, microbiomes are species-specific. Phylosymbiosis is observed at the order level, and size seems to be shaping microbiome composition across these individuals, especially in rodentia. And with that, I'd like to acknowledge my lab, my PI, Charlie Carsonell, in the Carsonell lab, and Sophia Green from RISD, who did the majority of the artwork for this project. And if you have any questions, please let me know.
Did you see any geographic signal across your plots?
Within species, yes, but it doesn't break the species. Can you hear me? Within species, yes, but it doesn't break that species-specific microbiome. So apparently, it can only change within that constraint.
How different is the diet between your smaller and bigger rodents? Do they feed on different things?
Someone else in my lab is studying that, and it doesn't seem like there's like distinct, like when you see the distinct species difference of the microbiome, you don't see a similar distinction in the diet. So you sort of see like a clone. If you do a PCA, it's somewhat like a clone.
That was amazing. And
thank you.
Do the rodents partition themselves in habitat in trees or fossorial?
They have different behaviors, and for example, I think one species is arboreal, so it's found in trees. So the good thing about this plot is that we're able to exclude these mammals, but they still have their native behaviors, right? So we're not controlling for their behaviors at all. But they do have distinct behaviors.
Great job. Does the accounting for phylogenetic distance between the substituted pairs of like microbes in like different communities?
Like a unified distance?
Yeah, like taking into account whether like, oh, this species is not these species are not here, these other species that are closer related versus distantly related microbes. Does that matter?
Yeah, it doesn't seem to matter in this group. It still seems like this correlation between size is real, it's the driving factor behind it. All right, thank you guys.